So let's begin with another session for today. The entitled as Mucoadhesive Micro NanoCarriers in Ophthalmic Drug Delivery System. We have a distinguished speaker for today's session, Mr. Sagar Pardeshi sir. Mr. Sagar Pardeshi sir is an assistant professor in Department of Pharmaceutics in SJIPR, Palghar from July 2022. Sir has completed his B Pharm in 2014 and M Pharm specialization in Sutics in 2016 from North Maharashtra University, Jalgaon. Currently, Sir is pursuing PhD from University Institute of Chemical Technology KBC, North Maharashtra University, Jalgaon. His key research area is ocular drug delivery. He is having 1.7 years industrial experience and 4 years of research experience. He has 11 international journal publications and 2 books chapters. With this brief introduction, I now request Sir to take over the session.
So very good morning and thank you for the kind introduction. The topic of my presentation is Mucoadhesive NanoCarriers in Ophthalmic Drug Delivery System. So in this presentation, we will go through the introductory part of the Ophthalmic Drug Delivery System, then anatomy of the eye. Thereafter, we'll see ocular absorption pathways. Next, we will cover mucoadhesive polymers which are available and the classification of Thank you for the kind introduction. My topic of presentation is mucoadhesive NanoCarriers in ophthalmic drug delivery system. So today we will cover the introductory part of the ophthalmic drug delivery system, then anatomy of the eye. Thereafter we'll see ocular absorption pathways, then mucoadhesive polymers which are available in the market, then classification of mucoadhesive polymers, and at last we'll see mucoadhesive NanoCarriers for ophthalmic application. So coming to the introductory part, as we know, eye, it is a complex organ and it contains significant barriers, like it has some anatomical and physiological constraints which are associated with the eye, like immediate drainage system. Thereafter, the low volume of the cul-de-sac cavity, that is 10 to 20 microliter. Then high sensitivity of the corneal layer. Then various physicochemical properties of the drug that also affect the absorption of drugs across the cornea. Thereafter, as we know, there are some drugs that are belong to different BCS class, like the BCS class 2 drug, which are low soluble and low permeable drugs. Sorry, highly permeable drugs. So as we know, in case of class 2 compounds, the rate limiting step for the absorption is dissolution. So in order to overcome the solubility issue, we have to go for nanonization. Again, In case of basis class 4 compounds, they are basically low solubility and low permeability drug. So in that case, we have to go for solubility enhancement as well as permeability enhancement. So for solubility enhancement, we can go for nanonization, and for permeability enhancement, we can go for lipid coating over the API. So accordingly, we have to decide different formulation strategies in order to improve the ocular absorption. Then this anatomical and physiological constraint like rapid turnover also lead to loss of the drug. Then smaller volume of cul-de-sac cavity, as I said, it is only 10 to 20 microliter. Thereafter, the corneal barriers which are associated with the eye may also lead to limited ocular absorption. And last one is the conjunctival absorption of the drug. So we will see one by one. So this is basically an anatomical structure of the human eye. So herein we can see the anterior segment of the eye. So basically eye is divided into two parts, that is anterior segment and the posterior segment. So anterior segment of the eye consists of, here we can see a cornea, then it consists of sclera, and it consists of a conjunctiva. Okay, so anterior segment consists of cornea, conjunctiva, and sclera, whereas posterior segment consists of inner layer of the eye, that is retina and lens. So in order to target posterior segment of the eye, your drug should cross the corneal rate, that is anterior segment. So the rate limiting step for the drug to get absorbed into the posterior segment of the eye is the corneal permeation. Okay, so your drug should permeable through the cornea, then and then it will reach into the posterior segment of the eye. So posterior segment consists of choroid, then retina, and vitreous bodies. So the presence of this corneal barrier, then rapid tear drainage and tear film turnover lead to poor ocular bioavailability. Now we'll see the different ocular absorption pathways. So drug which we used to instill into the eye, that is in the cul-de-sac cavity, basically absorbed by two pathways, that is corneal absorption and non-corneal absorption. So corneal route is the preferred choice of route for the drug which are belong to small molecular weight and are lipophilic in nature. So drugs which are lipophilic in nature and having a low molecular weight are generally absorbed through a corneal route, and drugs which are hydrophilic in nature and with a high molecular weight are usually absorbed through a conjunctival route. So once a drug is instilled into the eye, that is eye drop instilled into the eye, it will enter into the aqueous humor. Thereafter, it will distributed into the different intraocular tissues, and then it reach into the systemic circulation. Whereas drugs which are highly hydrophilic and large molecular weight will get directly absorbed into the systemic circulation through nasolacrimal duct. So the precorneal, the drug which are absorbed through corneal route have a limited ocular bioavailability, that is up to 1 to 5% of the total administered dose. Whereas drug which absorbs through the conjunctival or scleral route has higher ocular bioavailability, that is up to 50 to 100%. But the issue associated with the conjunctiva and scleral route is that drug is directly reaches into the systemic circulation through the nasolacrimal duct. So there is a chances of side effects. There are some ophthalmic formulation, means most of the ophthalmic formulation contain preservatives, and as we know, if the preservatives reaches to the systemic circulation, it may pose some serious side effects. So that we need to consider while developing the ocular dosage form. Then what is mucoadhesion? So muco, it is an inner layer of the mucosa. So as we can see here in the anatomy of the eye, so cornea basically consists of three layers. So first is a epithelium, second is stroma, and third is endothelium. So cornea basically consists of these three layers. In epithelium, it is lipophilic in nature, stroma, it is hydrophilic in nature, and endothelium, it is also lipophilic in nature. So the inner layer of the epithelium is nothing but the mucosa. So this inner epithelial cell is covered with a fluid and which consists of water and mucin. and generally this fluid is secreted by the goblet cells. So mucus fluid which is present over the epithelial cell contain protein, lipid, electrolytes, and mucopolysaccharides, and they act as a preservative and lubricant. Whereas adhesives are the substances which remain to a surface via hydrogen bonding, covalent bonding, or via electrostatic interaction. And if the adhered substance is a biological substrate, then it is a bioadhesion. And if the biological substrate is mucosa, then we call it as a mucoadhesion. Then what are the different mucoadhesive polymers which are available? So they are classified according to their source, according to the charge, then according to the solubility, and based on the type of interaction we can classify the mucoadhesive polymers. So according to the source, mucoadhesive polymers are classified as a natural and synthetic polymers. Natural polymers include chitosan, gelatin, carrageenan, pectin, cellulose derivatives like carboxymethyl cellulose, hydroxypropyl methyl cellulose, and sodium carboxymethyl cellulose. Whereas synthetic polymers contain carbopol, polycarbophil, polyacrylic acid, polyacrylates, polyethylene glycol with acrylate, acrylic acid, and polyvinyl alcohol. According to the charge, they are classified as cationic polymer and anionic polymer, that is positively charged polymer and negatively charged polymers. So they are basically aminodextran, chitosan, then PAC, carbopol, pectin, sodium alginate, sodium CMC, and CMC. So aminodextran, chitosan, they are positively charged polymer, while the remaining are negatively charged polymer. Then uncharged polymer, that is polymers which contain no charge, like hydroxyethylated starch, hydroxypropyl cellulose, polyethylene glycol, polyvinyl alcohol, and polyvinyl pyrrolidone. So these are the uncharged polymers. Then based on the water solubility, water soluble mucoadhesive polymers include carboxymethyl cellulose, HPMC, sodium CMC, HPC, PBA, PVP, and thiolated polymers. So basically thiolated polymers are the new generation mucoadhesive polymers. So in thiolation, what researcher did is they have converted the native or a core polysaccharide into the thiolated polysaccharide. So that hydroxyl group is replaced by a thiol group, that is SH group, and this SH group have tendency to interact with the negatively charged mucin which is present over the mucous membrane, and it form a covalent bond with the negatively charged mucin, and this formation of disulfide linkage between the polymer and the negatively charged mucin present over the ocular surface lead to higher mucoadhesion. Then water insoluble polymers examples are carbopol, polyacrylic acid, polyacrylate, polyethylene glycol, and methacrylate. Then based on type of interaction, covalent interaction based polymers are cyanoacrylates, then hydrogen bonding based are acrylate, carbopol, and PBA, and chitosan shows electrostatic interaction because it is a positively charged polymer, and as I said, ocular mucin, that is epithelial layer, is lined by the mucous fluid, and mucous fluid contains a mucin, which is a negatively charged protein present at the eye side. So positively charged chitosan electrostatically interact with the negatively charged mucin present over the eye surface. Now here we will see the interaction between the in situ gelling or mucoadhesive polymer with the mucous gel layer. So as we seen in the anatomical structure of the eye, the ocular surface contain inner layer, that is mucous layer, epithelial layer. So epithelial layer is covered by the lining of mucous fluid which contain protein, water, and different electrolytes. So the mucoadhesive polymers are basically water soluble polymers, or we can say as thiolated polymers are also a class of mucoadhesive polymers, and the polymers which contain strong positive or a negative charge may act as a mucoadhesive polymers. So the mucous gel layer containing the ocular mucin is of anionic character, and the structural characteristics of the mucoadhesive polymers are they are basically strong positive and negative charge which favors the ionic interaction. So groups which are able to form a strong hydrogen bond such as carboxyl group, hydroxyl group, amino and sulfate group shows mucoadhesion with the ocular mucin which is present over the ocular surface. Then carboxymethylation. So carboxymethylation, it is a derivatization approach of the core polymer. So you can convert any polysaccharide into the mucoadhesive polymer with the help of carboxymethylation approach. So carboxymethylation is basically involves Williamson ether synthesis approach. So this reaction involves the protonation of the hydroxyl moieties of the monosaccharide unit. So here we can see the hydroxyl moieties of the polysaccharide unit, which are reacted with the alkali hydroxide, that is NOH, to convert the alkoxide moieties. And these were further reacted with the monochloroacetic acid. And this through the SN2 reaction, we can get a carboxymethylated derivative of that polysaccharide. So what is the application of carboxymethylated polymer in ophthalmic application? So here I have taken example of chitosan. So chitosan, as I said, it is a deacetylated chitins, a cationic polymer which contain primary amino group. Okay, and this primary amino group is easily protonated at acidic pH. As it is easily protonated at acidic pH, it is highly soluble in acidic media. And as we know, the pH of the eye is basic, that is 7.4. If you put the chitosan solution into the eye site, what will happen? It may get precipitated at the eye site pH. That is why it is not feasible to add the chitosan solution directly into the eye. That is why researchers have developed carboxymethylated derivative of chitosan. So it is an amphiprotic in nature. Amphiprotic in nature means it acts as an acid as well as base depending on the medium provided. So carboxymethylated derivative, it has a wide range of solubility. It is biocompatible in nature. It is biodegradable, and it possesses low toxicity compared to pure chitosan. And it also impart improved mucoadhesion compared to the native chitosan. So a group of researchers developed dorzolamide loaded nanoparticles and polymer they have used, that is carboxymethyl chitosan. So when they have studied the mucoadhesion efficiency of this formulation, they found that the carboxymethylated derivative shows 37.76% of mucoadhesion with mucin, whereas the non-carboxymethylated derivative shows only 28.16% of mucoadhesion with the mucin. So from this we can conclude that the carboxymethylated derivatives has higher mucoadhesion because it has higher water solubility, and due to the higher water solubility, it can easily interact with the negatively charged mucin present over the ophthalmic site. Then next example is xyloglucan. So xyloglucan, it is a polysaccharide that is obtained through a tamarind seed. So xyloglucan with the 45% character removal ratio shows thermoresponsive gelling property. That is, it is solution, it remains in the solution form at room temperature, whereas if we increase the temperature up to 35 or 37 degrees Celsius, it converts into the non-flowing gel. But the issues associated with the xyloglucans are it has a dull color, unpleasant odor, it has limited solubility in cold water, and it contains some water insoluble components that limits its applicability in drug delivery. So researchers have developed carboxymethylated derivative of xyloglucan. So this is the derivatized derivative of xyloglucan, which is formed by the Williamson ether synthesis approach, and this derivatization exposes the polysaccharide network of that xyloglucan that result in the higher viscosity, improved solubility, and enhanced mucoadhesion. So again, a group of researchers have developed dorzolamide loaded carboxymethylated xyloglucan polymer, and what they found is that the in situ gelling time, that is time required to convert a solution into the gel, is reduced. That is, in situ gelling is observed within 1 minute, and the formulation shows prolonged drug release, that is up to 10 hours. So this is the application of carboxymethylated xyloglucan over the native xyloglucan. Then thiolation approach, as I said, these are new generation mucoadhesive polymers called as thiolated polymers. So basically they are prepared by esterification reaction of the hydroxyl group of the polymer with the thioglycolic acid in presence of HCl. So here we can see SH group is attached to the backbone of the pure polysaccharide polymer. So these SH groups are interact with the mucin which is present over the ophthalmic surface and form a covalent bond, that is strong bond between the mucin and the polymer. And due to the formation of the strong bond, there is an enhanced mucoadhesion compared to carboxymethyl derivative. So in case of carboxymethyl derivative, there is a hydrogen bonding and electrostatic interaction. But in case of thiolated polymer, there is a covalent bonding. And as we know, covalent bonds are the strongest bond. And that is why thiolated polymers are called as a new generation mucoadhesive polymers, because they show highest mucoadhesion compared to other polymers. So these are some examples of thiolated polymers. So native hyaluronic acid, it is an anionic non-sulfated linear polysaccharide of natural origin, and it has ability to retain water through hydrogen binding. Thus, it is commonly used to relieve dry eye syndrome and also in the treatment of wound healing. But researchers have modified this hyaluronic acid into the thiolated hyaluronic acid, and this thiolated polymer shows enhanced corneal residence compared to hyaluronic acid due to the formation of covalent bonding between the mucin. And it shows prolonged drug release because thiolation imparts rigidness to the polymer, and due to the rigid structure and due to the covalent interaction between the polymer and the mucin, the form polymeric structure provide a diffusion controlled drug release of the polymer. That is why there is a prolonged therapeutic effect we can achieve with the help of thiolated polymers. Then they have checked the revitalization potential of this formulation and compared with the native polymer, and they found that this synthesized cross-linked hyaluronic acid film shows 99% of the revitalization at the end of 48 hours, while the control formulation showed only 86%, which is estimated through fluorescence uptake study. So here we can see that this covalent interaction enhances the residence time of the formulation at the eye site. Furthermore, it imparts the rigidity to the native polymer. Then it has also increased the revitalization rate of the polymer. Then next example is pectin. So pectin, it is a anionic polymer, that is negatively charged polymer, which is biocompatible, biodegradable, non-toxic, and it has a high molecular weight. And the high water solubility of the pectin allows rapid hydration, swelling, and erosion that reduces its mucoadhesion potential and the ability to control the drug release. That is why to overcome this issue, researchers have developed thiolated pectin, and it shows enhanced mucoadhesion due to the formation of covalent bond. Then ex vivo studies, that is permeation study across the corneal layer, shows that improved mucoadhesion, that is compared to the pectin, thiolated pectin shows enhanced mucoadhesion. Then again, thiolated pectin containing beads had a percent swelling 28.4, while non-thiolated pectin shows percent swelling of 66.9%. Then what are the recent strategies that we can incorporate in conjunction with the mucoadhesive polymers? So first is nanoparticles. So nanoparticles are basically sub-nano-sized colloidal particles having particle size in the range of 10 to 100 nanometer. So in this research, a group of researchers have developed pilocarpine-loaded PLGA nanoparticles. So PLGA nanoparticle, it is basically a controlled release polymer, and pilocarpine, it is an anti-glaucoma drug. So glaucoma is a condition where excessive fluid is built in the eye, and that may lead to the vision loss. So in order to treat the glaucoma, pilocarpine is incorporated into PLGA nanoparticles. So the intention of the use of a PLGA nanoparticle is that to provide a controlled drug release, because pilocarpine it has a short half-life, and as we know, the compound containing short half-life are rapidly excreted from the body. So in order to maintain the effective therapeutic concentration level in the body throughout the treatment, they have used a controlled release polymer, and this controlled release polymer is further coated with the chitosan, that is positively charged polymer. So here, why they go for a positively charged polymer coating over the PLGA nanoparticles? In order to improve the mucoadhesion. As I already said, positively charged polymers are strongly interact with the negatively charged mucin present at the eye site. That is why they have used the coating of chitosan polymer over the PLGA nanoparticles. So they have performed the turbidimetric measurement and the zeta potential measurement of this developed formulation. And here In the first figure, we can see what they did. They have incorporated this nanoparticle in the water and measured the zeta potential of that formulation. And zeta potential determine the surface charge of the particles. Okay, so the surface charge of the particles is around plus 55 millivolt. When same nanoparticles when incorporated with the mucin, that is ocular mucin, and after 1 hour of incubation, zeta potential was reduced up to 30 millivolt, that is plus 30 millivolt. So reduction in the zeta potential is due to the interaction of the negatively charged mucin and the nanoparticles, positively charged nanoparticles. That is why there is a formation of complex between the negatively charged mucin and positively charged cationic polymer, and thus there is a reduction in the zeta potential. So from that we can conclude that there is a mucoadhesion, there is an interaction between the mucin and the positively charged polymer. And once the incubation time is increased, we can see further decrease in the zeta potential. That is, more chitosan is complex with the mucin molecules, and that is why zeta potential is reduced. And same thing we can see in the turbidimetric measurement. So 0.1% mucin and non-coated mucin shows less turbidimetry, whereas the formulation, that is chitosan 1 and chitosan 2, higher turbidimetry. So turbidimetry is increased due to the formation of complex between the negatively charged mucin and positively charged polymer, and there is a formation of turbidity or the formation of complex. We can conclude with the help of zeta potential and turbidimetric measurement. Then next is microparticles. So microparticles are basically colloidal drug carrier having size in between 0.1 to 100 micron. And for ophthalmic application, the particle size should be less than 10 micrometers so that it should not cause any irritation to the eyesight. So a group of researchers have developed a elastin loaded chitosan alginate mucoadhesive microsphere, and they possess a mucoadhesion efficiency up to 65%, and also showed a controlled drug release up to 8 hours. And the eye scratching study in the animal showed less scratching instances for microsphere up to 8.83 compared to the positive control that shows scratching instances of 22.66, which clearly indicate that the improved therapeutic efficacy. The same we can see from the graph. So negative control, it has scratching instances of around 20, and here we can see elastin chitosan alginate microspheres showed less scratching instances, that is up to 10. Then next is liposomes. So liposomes are basically microscopic artificial vesicles consist of one or more phospholipid bilayers, and their size varies from 10 nanometer to 1 micrometer or greater. So in this study, researchers incorporate the developed liposome into the gelling matrix. So they have compared the permeation ability of the free liposome with the viscous formulation containing 16% of the viscosity building polymer and another viscous formulation containing 24% of the viscosity building polymer. So through the fluorescent uptake study, they found that the free liposome at the end of 10 hours, we can see complete black spot at the end of 20 minutes. So drug is available across the cornea for 10 minutes only. But in case of viscous formulation, as the viscosity increases the residence time of the drug at the corneal site, we can see viscous formulation containing liposome shows presence of drug at the end of 60 minutes also. And same in case of gel formulation containing 24% of viscosity building polymer. So from that we can conclude that the hydrogels or in situ gels or viscosity building polymers are also contribute in the enhancement of mucoadhesion and the permeation of the drug. So conclusion. So as we know, to improve the bioavailability and to get a desired drug at the eye site, we can use a mucoadhesive carriers. The modification of the core polymer by carboxymethylation or thiolation is a promising approach in order to improve the residence time of the formulation, to improve the permeability of the formulation and subsequent bioavailability of the formulation. So mucoadhesive polymers are basically can be combined with the nanoparticulate system like nanoparticle, microparticle, liposome, neosomes, to deliver all kind of drug, that is this is class 1, class 2, class 3 and class 4 drug for ophthalmic application. And the selection of this mucoadhesive polymers and its derivative with suitable nanoparticle system depend on the desired effect required, and accordingly selection has to be made to develop an effective drug delivery system. So these are some references. Thank you so much.
Good morning, everyone. It's a great privilege and honor to propose the vote of thanks for today's session. I extend my gratitude to our eminent speaker, Mr. Sagar Pardeshi, sir, for his relevant address on mucoadhesive micro nanoparticle carriers in ophthalmic drug delivery system. Sir has focused on various points like what are the routes of absorption for topical applied ophthalmic, what is mucoadhesive, what are the mucoadhesive polymers classification, what are the examples of carboxyl methylate polymers with its property, also explain nanoparticles, microparticles, liposomes, and comparative and explain the comparative study with an marketed formulation. Our sincere thanks to our chairman, Mr. Albert D'Souza, sir, for his constant encouragement, and thanks to the Mrs. Alvina D'Souza, ma'am, Secretary of Eldel Education Trust, all the other dignitaries of the management. A special thanks to our principal, Dr. Savita Tauro ma'am, for eminence guidance and support. My deep sense of appreciation and thanks to all the participated who attended this webinar with the great willingness. Feedback will be uh, feedback will be provided in your respective WhatsApp group. Kindly fill it so that it will be helpful us to improve our program. Thank you all. Have a wonderful day ahead.